Thanks, Greg. Hi, everyone, and thanks for coming. I'm Adam, and today Yoshiki and I are going to talk to you about Qiita and Emperor, which are two Python projects we've been developing in the Night Lab for conducting microbial ecology analyses, which is a subject that not everyone is familiar with. So here we have life as we know it, and it's the tree of life that shows all three domains, including bacteria, archaea, and eukarya, and within each domain, representative genera. Now, if you look at the bottom left corner of the eukarya clade, you can see three genera that represent all of the macroorganisms, all the plants, animals, and fungi that you're used to dealing with on an everyday basis. Now, the rest of the tree, that is to say, the vast majority of life on the planet, is microbial in nature. And it's not like they're rare by any stretch. They're actually pervasive, ubiquitous, essential components of all ecosystems. And if we look at some terrestrial ecosystems like Arctic or desert or hot springs or jungles, we're definitely going to find microbes in all of these environments, and they're actually performing important functions and determining the geochemical composition of the biosphere. So these are some environments you might be used to thinking of, some biomes you're used to considering. But you might not immediately think of the ecosystems in and on the human body. So you have about 10 trillion human cells in your body. You have 100 trillion microbial cells in your body. This amounts to actually about 4 to 5 pounds of your mass. The reason it's a low number like that is that the cells are actually quite a bit smaller than your cells. But more importantly than the cell count is what these cells are actually able to do. So you have 20,000 or so human genes that basically determine what kinds of biochemical functions you can perform with your genome. But there are 2 million unique microbial genes that are in those organisms. So it vastly increases the repertoire of biochemical operations you can do. So we know these microbes are doing important things in the body, like helping us metabolize food, synthesize vitamins, and training our immune system. So it's not like they're just along for the ride. They're actually an integral part of you as a person. So it really behooves us to understand as much as possible about these organisms in order to try to manipulate them for health benefits, for example. So the kinds of questions we can ask about these microbes are: who are they, what are they doing, how much are they doing it, and how are they doing it? And that's basically summarized in these questions on the left side. And we have to use a variety of different omics techniques to address these questions, such as 16S amplicon analyses, metagenomics, transcriptomics, proteomics, and metabolomics. Now, this is going to be a ton of data, and it can be a little bit like drinking from a fire hose, because even just one of these techniques, metagenomics, can result in terabytes of data. Now, if you wanted to analyze samples using more than one omics technique, you have to combine that data, and when you consider all the other metadata, integrating it can provide a lot of technical challenges related to the organization of the data, the storage of the data, the standardization of the metadata that'll allow it to be easily reused. And then even when you've done all that, you still have to address the problem of actually performing computation on this massive data set. So you have to consider how and where you're able to do that. And finally, once you've generated results that you want to publish, depositing that data can actually be quite a headache. So what would be great is if there were a tool that allowed this kind of a workflow, where once the scientist generates all this data, the technical burden from data acquisition to the generation of biological insights is minimized. So we're developing Qiita, and this is a platform for conducting this kind of multi-omic analysis across several studies. So what I'm showing you here is kind of a diagram of one of the central data models in Qiita. So it's kind of a study concept that encapsulates the study metadata. So that includes things like who wrote the study, what's the title of it, what's the abstract of the study, et cetera. And then you're also going to have a sort of sample metadata. So for soils, you might have pH or something like that. And then preparations of those samples for different omics techniques. And then together with all of that, you have the primary data that you're getting off your instruments, as well as the results you're generating from that data. So we encapsulate all that in this central data model. So on top of that data model, we have a couple of different interfaces. because we're trying to minimize these technical burdens, the main interface for Qiita is a browser-based graphical user interface, which is portrayed here on the left. But we also have command line interfaces for those more technically minded. And so you can deploy Qiita on your laptop. It's all open source. All of Qiita and all of its dependencies are open source. You can put it on your laptop. You can put it on a desktop machine if you want to gain the kind of organizational benefits that Qiita can give you. But the more common use is as a centralized multi-user system. So there's one such system running at the Broad Institute, and the Knight Lab and the Qiita developers maintain a deployment of Qiita on UCSD hardware at qiita.microbiome. So one of the benefits of using a centralized system, and we recommend this one, is that you get free storage and computation on UCSD hardware, which is a nice feature. Also, as I mentioned, data deposition in public repositories can be a real headache, and so we streamlined that process, and really, once you have it curated and approved by the moderators, it's a single click of a button to get it deposited onto EBI, and you get your accession number that you can then publish. Furthermore, you gain access to all the other public studies that are deposited on the system, so you can actually look at your data in the context of all the other public data. And so that's running on UC San Diego's hardware. So what I'm going to do right now is show you how you can do a meta-analysis on that live system. So here in this browser, I pulled up qiita.microbiome, and after you create a user account, which is just a simple and free process involving email verification, you see this front page. And so this is running on a web server, on Tornado's web server, or using Tornado as a web server, and the processing pipeline that it implements is all based on NumPy, SciPy, and Chime, which is used for the main analytical pipeline for 16S data. So if I go into the studies and view the studies that are currently available on the system, what you'll be presented with is a subset of, by default, 10 of the total 187 studies that are available for analysis. Now, let's imagine for a second that I'm a microbiome researcher interested in soil samples. So I'm going to search and select a subset of the studies for consideration where I'm going to use a standardized metadata field called env-matter, which controls, so env-matter is an example of a standardized field from the ENVO ontology, which, so again, we manually curate a lot of these so that they adhere to these ontologies so you can search across studies. So I'm going to say that the env-matter includes soil. So I'm going to shrink it a little bit again. So now, down here, it's showing us that we've limited it from the original 187 down to 50. So this is a more manageable number, but if I were a soil microbiome researcher, I'd be familiar with Noah Führer's work, and so I might want to filter this current set of results based on some of the study metadata, something in the title, something in the abstract, or the author. So I'm going to filter the results to include, so I'm typing Führer into that filtering box. And now I'm down to just 5 studies. And so this is a more manageable set of studies on which I might want to conduct a meta-analysis. So by clicking on these icons next to the titles, I can pull up the abstracts so I can get a better idea of what the study is about. Or if I, sorry for all the zooming, it's just resolution. But if I then click on the PubMed ID that's associated, it pulls up the entry of the paper associated with that study in PubMed, so I can get a much more detailed explanation of what the study is all about. So let's go ahead and do a meta-analysis of a couple of these studies. So as an example, there's one study here called Soil, Bacterial, and Fungal Communities Across a pH Gradient in Arable Soil. So that's kind of a good backdrop to compare things against, because it spans all soil types and provides a pH gradient. Next, let's select this last study called Soil Bacterial Diversity in the Arctic is not fundamentally different than that found in other biomes. So by clicking the Add to Analysis button, you can see a little notification come up notifying me that 52 samples were added to my current analysis. And right now I have 79 selected from these 2 studies. So if I go to the analysis menu up here and select create from selected samples, I'm brought to a page that summarizes what I've selected for analysis. And so you see here the 2 studies that I just selected. So what we're seeking to do here is to investigate the claim that the Arctic samples do not differ significantly from other terrestrial biomes or other soil samples in other contexts. So if I hit create analysis, I'm brought to a page where I just have to enter a simple analysis name. So it's going to be Sci-Fi 2015 demo. And I hit create analysis. And immediately I'm presented with just a few options, much simplified a much simplified interface where I just have to select what kinds of analyses I want to run and the visualizations I want to generate. Because this would take about 5 to 10 minutes to run on this particular data, we don't have time to do it right now. I'm just going to say that we're going to generate taxa summaries and beta diversity plots. And then I'm going to hand it over to Yoshiki, who's going to discuss the kinds of visualizations you can generate from this data using some pre-computed data on this exact data that we ran last night.
So like Adam said, I'm going to be switching more over to the visualization capabilities of Qiita. And the most relevant one here is the one powered by Emperor. And so to give you a little bit of background, Emperor is this Python tool that we started development on a couple of years ago, where we really just have these this small list of requirements. We needed this to be very portable, so it couldn't run on a server to actually present the data, or it couldn't require you to install an application on your computer to actually be able to open these files. And we also needed it to be able to use the metadata that's associated with all of your samples in a way that you could modify your plots and you could change different attributes based on these values. But I think the most important part is that we needed these to be interactive. We needed these changes to be able to happen as the user just changed or interfaced with the graphical user interface. And so I'm going to go back into Qiita, and I'm going to go to my list of previous analysis. And so this is the analysis that we ran last night and a few of the ones that we rehearsed on. And what I'm presented with here is just the results that come out of Qiita. And so as Adam mentioned, one of them is going to be taxonomic summaries. And so this is just a summary of the composition of the microbial communities that are represented in each of these samples. And this is done at different taxonomic levels, but I think the more interesting part here is if we go to the beta diversity plots. And beta diversity, just very quickly, is a technique that microbial ecologists use to assess how similar or dissimilar are two communities. That meaning how closely related is the microbial composition of two different biological specimens. So each of these spheres here represents a sample, and depending on how close they are to each other is how similar their microbial composition is. So we can, for example, color by the title of the experiment. And so we can select that, and we see that we have two titles, and I'm just going to quickly zoom into this. And these are the studies that Adam selected in the previous study list, but the spheres are actually kind of small, so we can change these really quickly and just make it clearer for you to see what's going on. So to verify the claim that Adam was wondering about of whether or not soil communities are similar regardless of whether they come from regular environments or Arctic environments, we can, for example, take a look at the pH metadata field. And so now we see that it's a list of numeric values, but it's not really very useful as a discrete color map. So we can go ahead and change these into, for example, an orange to red gradient. And what we are able to see is that indeed samples that have a lower pH clustered together with samples that have with other samples of the similar pH. And we see this really nice gradient on the first principal coordinate axis, which in this context is kind of a good thing that you would expect to see. So just going back, I also wanted to mention that by adhering to these very minimal list of requirements, we've been able to integrate Emperor in a variety of platforms. And as you just saw, we were able to put it in within Qiita. It's been very successful at being integrated in the QIIME pipeline. Recently, it was integrated in Illumina's base-based platform for analysis of sequence data. And at the Night Lab, we also used it to deliver these, results for participants of the American God project, which is this crowdsourced and crowd-funded project where basically you send us a biological specimen and then we send you back a report of how the microbial communities are structured in these biological specimens. And the cool thing is that 3 out of those 4 figures were generated with Emperor and were being delivered to these participants. Just to wrap up, I wanted to say that with Qiita, we're still in an alpha stage, and there's still a lot of things that can and will improve as we continue development on it. And that for now, we only really have a 16S pipeline, but we've set the foundation so that we can expand onto other omic fields or omic data types. And that for Emperor, we are on a beta status at the moment, but we're approaching a stable status by the end of the summer. And we would really like to hear any use cases you may have with any of these tools. And just that we also adhere to all the standards that we love and like from the scientific Python community, such that it's easier for you to come and contribute. And with that, I would like to thank you for your time. Right, so the question is, does Qiita allow you to keep samples private? And the answer is yes. We allow you to keep your samples private while you're analyzing them and while you're preparing your manuscript. But really, we try to encourage people to, once you've published your paper or your data set, that you make these public such that other users can come and grab the study quickly and just be able to integrate it in another analysis. So the question is, what data formats can you get from Qiita and at what stages? And the short answer is you can get process tables like biome tables that is a genomic standard consortium file format. The multiplexed and quality controlled sequences, and you can also get the mapping files that are compatible with QIIME and or other sort of just very basic tabular data formats in case you're not really necessarily working with QIIME, but would still like to have a look at the data in, say, MATLAB or R.